Sahu, and she will be speaking on design and development of tractor-mounted intercom intra-row weeder. Could you please have a round of applause for the session chair? We are having eminent scientist Dr. Ramajayam Devarajan from IISWC RC Koraput. Let's welcome sir with a round of applause. We are having our esteemed co-chair Dr. P. Srinivas, esteemed principal scientist ICAR IAHR. We are having two of our rapporteurs for the session, Dr. Santanu, eminent scientist, Chase Bhuvaneshwar, and Dr. Hare Krishna Swami, eminent scientist from Botanical Survey of India. A louder round of applause for all the esteemed panel. Moving ahead with the first presentation, which will be on design and development of tractor-mounted intercom intra-row weeder by Sarmistha Sahu from CUTM Bhubaneswar. Let us begin the parallel session virtually. Sarmistha, could you please have your slides?
Yes, madam.
You are having 10 minutes of time. It is 11:20, almost 11:30 right now. So accordingly, we will begin. Over to you. You are having
Thank
10.
you, madam.
It is 11:20, almost
Uh,
right now. We will
is
begin.
it visible?
Yeah, so visible. You can start. 10 minutes is the time.
Yes. Thank you, sir. Okay, sir. Good morning, everyone. I'm Sadmista Sahu. I'm doing my PhD in Centurion University and Technology Management, Parlakhamdi, Odisha. I have completed my graduation and MTech from OUAT Bhubaneswar. From today, I'm going to present topic design and development of a tractor mounted intercom intra-row weeder, which is my PhD topic. So, in India, weeds are one of the major biological constraints that limits the crop productivity. Weeds in the crop field compete for soil nutrients, soil moisture, sunlight, space, water, and other ecological factors. In row crops, the field is invaded by inter and intra-row weeders. The weeds in the intra-row zone reduce the crop yield up to 33% or more. So, in this pie chart, we can see like the weeds is more affected by the weeder. So, from others and disease, insects affect. So, 45% is affected by the weeds. So, from the weeds, both of them are either inter-row or as well as intra-row. So, inter-row is the weeds which is present in between the row, and intra-row means the weeds is present in between two crops. So, there are many methods of weeding like manual weeding, mechanical weeding, and chemical weeding. So, weeding method, we do the weeding manually, as well as in chemical weeding, we do the spraying of pesticides by using aerial application or by sprayer. So, which again enhance the which increase the pesticides and which is very dangerous for us now. So mechanical weeding is the best method of removal of weeds. So there are many types of weeder already available in market like tractor or inter-row weeder, some robotic weeders, power weeder, and tractor and finger weeders are also there. So here in this tractor and finger weeder, we can see it is removing the weeds both from inter the weeds and some percentage of in between the plant. It also remove the weeds. So in my topic, in my project, I am willing to remove the weeds from the plant. In this diagram, we can see the inter row weeds. So here, these red color weeds are the intra row weeds, and green weeds are the inter row weeds. So here, one weeder will be sufficient which remove the weeds from both the inter as well as intra row. So the design objective is first to design a CAD model and develop a tractor-mounted inter and intra-row smart weeder to develop a sensor-based guidance and control the system for developed prototype, then to study the performance evaluation of the developed prototype weeder in the laboratory as well as field condition. So these are the work progress I will have. Like first of all, I have to take the what are the types of crop I want to work for. Then what is the row spacing and plant height and other crop parameters I have to take. Then the design consideration, the 3D model. Then I have to test the efficiency at a soil bin. Then I will incorporate the machine with a tractor, then do the field evaluation. So these are the working principles. First, for designing the intercom intra-row widths, these following parts needs to be developed, like so main frame, intra-row sweeps, intra-row rotating shaft, then bearing, intra-row rotors, knives, rotating shaft for actuation system and power transmission system. So here this is the design of the project, that is design of the tractor mounted inter and intra-row weeder. In this, the first inter-row sweeps, these will be sufficient for removing the weeds from in between the two rows. Then the intra-row rotating shafts, which is just behind the inter-row sweep, which will be in a rotating manner, it will remove the weeds from the plant, that means in between the plants. In this diagram, we have seen that. That means here, this U-shaped part, it is the path showing of the movement of the rotor. So here this rotor will rotate, so it will remove the weeds in between the two plants. Then this is the power transmission system and mainframe. So the actuation system will be done by the sensors. So there will be ultrasonic sensor and proximity sensor, which will detect the plant distance, as well as it will detect the plant dimension, so that it will detect it, as well as it will help in actuating the inter-row rotating shaft. So these are the results. So this is the developed prototype. Here, these are the inter-row, and these will be the intra-row application with this. So this is the power transmission system, which will be taken from a tractor. So one gear is there. So it will take the power from the PTO shaft of the tractor, and it will rotate the rotors. And for the front one, that is inter-row sweep, it doesn't require any power transmission. It will just pulled by the tractor. So these are the parameters I have taken. Field performance, like crop, I have taken the okra, like spacing will 45 and to 30 centimeter. The depth of operation 5, tractor forward speed 2.5, rotor peripheral speed 1.75 meter per second, rotor speed 280 RPM. Here the weeding efficiency is very good, like 96% weeding efficiency. Effective field capacity is 0.34, fuel consumption 4.6 liter per hour. So here, the material which is used for the fabrication is mild steel. So these are the overall dimension of this tractor mounted intercom intra-row weeder. So I sincerely acknowledge the support and facility provided by the Department of Agricultural Engineering, Centurion University of Technology and Management for carrying out the research work. I also express my sincere gratitude to my advisory committee member and technical staff for their valuable guidance and continuous encouragement throughout the study. Thank you, sir. So this was my presentation. So this was my presentation.
Yeah, you are well within the time. Is there any provision to ask question among the online
Yes.
participant also?
Yes, sir. Please.
Anyone present in the online would like to ask any question to Sarmishta? To ask question among the online participant also?
Yes, sir.
Anyone present in the online would like to ask any question to Karmishta? Ask question among the online participant also.
Yes, sir.
Okay. If not, shall I ask a question, Sharmista?
Yes, sir, please.
Okay. So if I go for we decide, or if I go for manual weeding, if I go for your own this machine, so which one will be economical? If I and what is the cost for your machine? If I use your machine, what will be the cost of weeding per hectare? This machine. So which one will be economical?
Okay, sir.
And what is the cost for your machine? If I use your machine, what will be the
Sir,
cost
for
of weeding per hectare? This machine.
actually, I'm getting echo of.
Yeah, that's what I don't
What
know. We are fixed, for your machine. If I use your machine, what will be the
Yes, sir. Sir, for manual weeding, it is time consuming, but the efficiency is 100% because we can see the weed, we can remove the weed, but time is requirement is very high. Like for 1 hectare of area, 48 number of farmers need to work for 1 day for removal of weeds. So if we like give 500 rupees of labor cost for one farmer, you can calculate like how many amount will be used in 1 day for removing weeds from 1 hectare. But in this machine which is developed here, the field efficiency is 0.34 hectare per hour. So here field capacity is 0.34. So it will take in 1 day, it can remove, like if it is operate for 8 hour per day, that means the working hour is 8 hours. So if 5 6 hour operation, it can remove all the weeds. Here the cost of the machine, it is still in progress, sir. So design part and development part is still going on. So the cost till now I have not calculated, but it will be around 1 lakh rupees. So if somebody like farmer will purchase this for inter as well as intra row weeding weeder, so it will be enhance the productivity as well as it will reduce the labor cost as well as it will also reduce the cost of operation and timeliness operation also, because if we take the help of labor, it is again time consuming. So crop harvesting period also achieve during this process, so we can do this.
cost of cultivation? Why? Because definitely any farmer or any entrepreneur who want to take your technology will ask what will be the cost saving by this machine. So that is very much important. So you can work it out. What by taking into cost of this equipment, how many years you can keep it by putting a future cost, all those things. It is possible to work out. Machine. You can work it out. Okay. So you can work it out. What by taking into cost of this equipment, how many years you can keep it by putting a future cost, all those things. It is possible to work out. Machine. You can work it out. Okay. Did you get my point? Work it out. What by
Yes,
taking
sir. Yes,
cost
sir.
of equipment,
Sir, this is my work now. Now also it is going on. So that's why this is the last part is the cost economy part. So that is the last objective. So till now I have not done that, but I will definitely do that, sir. You are not audible, sir. Hello.
Is it audible, Sarmishta?
Yes, now it is audible, ma'am.
yeah. No, my question is up to what percentage of land slope your machinery can work?
Is it audible, sir, Mr.?
Yes, now it is, ma'am.
Yeah, no, my question is up to what percentage of land slope your machinery can work?
Is it audible?
Sir, generally, all these, like
Yeah, no, my question
whatever the
is
crop
up to
we
what
have.
percentage of land slope your machinery can work?
Is it audible?
Sir, machine is designed to work in a plain land, sir. Whatever there should not be any slope.
Land slope, your machinery can work.
Is it audible?
Okay, thank you.
Sir, machine is designed to work in a plain land, sir. Thank you, sir. Thank you, everyone. This was my time for presentation. Thank you all. Ma'am, you are not audible.
Anushka from Banaras Hindu University, Varanasi.
Ma'am, you are not audible.
Anushka is there?
Sharmista, you can unshare your PPT first. Kindly unshare.
Banaras Hindu University, Varanasi.
Ma'am, you are not audible.
Sir, Mr. You can drop your slides.
Sharmista, you can unshare your PPT
Let's connect
first.
to Anushka. Is she there?
Kindly
From
unshare.
BHU Varanasi. Varanasi.
Ma'am, you are
Now,
not
among the waiting list, you can see
Sharmista,
that
you
there
can
we
drop
can pick up.
slides.
I think it is. Okay, I think she may not be
Let's
there.
connect to Anushka. Is she there?
Kindly unshare.
BHU Varanasi.
Now, among the waiting list, you can see
BHU
that there we
Anushka
can pick up.
Chandiki.
I think it is. Okay, I think she may not be there.
Connect to Anushka.
Sayani Das is there.
BHU Varanasi.
Anushka, Sayani
Yes,
Das,
sir. I am present.
Sayani Das. Okay, you please go ahead. You can
Okay,
share
sir.
your.
Okay, sir.
Sayani Das is there. So she will be presenting
Yes, sir.
climate
I am
resilient
present.
integrated farming system modules as a sustainable strategy for nutritional and livelihood security of smallholder farmers. So she will be presenting.
May I kindly confirm whether I am audible and the slides are clearly visible?
Yeah, you are audible. Your slides are very clear. You can go
Thank
ahead.
you, sir.
It's also moving. Slides are moving.
Thank you, sir. Good morning, respected chairperson and esteemed fellow presenters. I am Shaini Das, PhD scholar from the Department of Agronomy, Bidhan Chandra Krishi Vishwavidyalaya, West Bengal. Today, I will present my work on climate resilient integrated farming system module as a sustainable strategy for enhancing food, nutritional, and livelihood security of smallholder farmers. This study addresses one of the most pressing challenges of Indian agriculture: how to make smallholder farming productive, resilient, and sustainable under increasing climate stress. So, Indian agriculture is dominated by small and marginal farmers, and land fragmentation has made horizontal expansion unfeasible, requiring productivity enhancement through vertical intensification within existing land resources. This makes intensification within the same landholdings not just an option but a necessity. Current farming systems are mainly monocropping and single enterprise based, with poor integration of crops, livestock, and allied components, leading to inefficient use of land, water, nutrients, and biomass. As a result, potential synergies among enterprises remain largely untapped. High dependence on external inputs increases carbon emission, while limited biomass recycling reduces soil carbon sequestration and degrades soil health. This further threatens long-term sustainability and ecosystem service. Existing systems show low resilience and weak buffering capacity against climate extremes, especially in fragile and coastal ecosystems. Yield instability and income uncertainty are becoming increasingly frequent. Monocropping provides seasonal employment with low labor absorption and causing income instability among smallholders. This directly affects livelihood security and rural well-being. Thus, present study farming system fail to meet the need of smallholder agriculture. Therefore, smallholder specific vertically intensified and integrated farming systems are
essential to enhance productivity, carbon efficiency, climate resilient, and year-round employment. These form the conceptual foundation of the present study. So, what is integrated farming system? Integrated farming system is a whole farm management approach based on the principle that there is no waste and waste is only a misplaced resources. IFS aim to recycle on-farm resources, reduce external inputs, sustain farm income, ensure nutritional security, and minimize environmental pollution while maintaining the multifunctionality of agriculture. In essence, IFS transform farm into circular self-regulating agro-ecosystems. This enhances system efficiency while simultaneously addressing economic, environmental, and social dimensions of sustainability. IFS integrates crop, livestock, horticulture, poultry, fishery, etc., forming a biologically linked and self-supporting system. The strength of the IFS lies on individual component but in their functional integration. With this background, our objectives were to study the productivity and profitability, to assess the resource recycling, to study the employment generation, to estimate GHG emission. Together, these parameters capture system performance in a holistic manner. With this perspective, the IFS module was evaluated under the 6th cycle of AICRP IFS 2022 to 2023 at Odisha University of Agriculture and Technology. The model covered 1.12 hectares. There were 5 major modules which includes cropping system, horticulture, dairy, poultry and composite fish culture. Each components are designed to support others through internal resource flow and nutrient cycling. This model was designed keeping smallholder feasibility and climate adaptability in mind. Enterprise proportions were optimized to ensure both food security and regular cash flow. Coming to the results, the integrated system achieved a total productivity of 37 tons rice equivalent yield per hectare. Dairy emerged as a major contributor, followed by horticulture. Poultry and fishery ensures quick and seasonal income, while crops provide food and fodder security. This clearly demonstrates that the system stability increases when dependence shifts from seasonal crops to vertically integrated year-round biological enterprises. This diversification reduces risk while strengthening overall system resilience. Economically, all enterprises are profitable with benefit cost ratio exceeding 1.6. The system generated a net return of approximately 4 lakh per hectare, far superior than conventional system. More importantly, the model ensured income continuity, thereby protecting farm families from income shocks and distress borrowing. This demonstrates the economical robustness of integrated farming system compared to monocropping farming. A key strength of the system was efficient waste management and resource recycling. This creating a closed nutrient loop that minimize waste, reduce external input dependency. Almost 15% of the input cost is contributed by the recycled products. This circular nutrient flow significantly improved nutrient use efficiency. This model created 399 man days of work annually because it integrates multiple enterprises. Dairy contributes over 50% of the labor since daily activities like feeding, milking, and managing animals require consistent effort. Horticulture and crops add more seasonal work. This integration ensures continuous employment, allows farmers to develop diverse skill, and strengthens the local community socially and economically. Such employment continuity is critical for reducing seasonal migration. From an environmental perspective, the system delivered an exceptional outcome. Despite emission from the dairy component, the model achieved a carbon negative balance of minus 289 kilogram carbon dioxide equivalent per hectare per year. This highlights the potential of integrated farming system as a climate smart agriculture strategy. Coming to the next slide, horticulture showed the highest energy use efficiency. Fishery recorded the highest energy productivity, and the integrated system achieved an overall efficiency of 2.85, highlighting the advantages of enterprise integration. This confirms that integrated system optimize energy utilization across enterprises. Over the 6 years, the model improved soil health significantly, water holding capacity and organic carbon increased, nutrient availability improved, and bulk density decreased, indicating better soil structure and fertility under both horticulture and cropping system. These improvements reflects the long-term ecological sustainability of the system. The long-term sustainability analysis revealed a sustainable yield index of 0.74 and a sustainable value index of 0.43, confirming that the system delivers not only high productivity and profitability, but also consistent dependence and resilient performance over time. This confirms both stability and reliability under variable conditions. Coming to the conclusion, the IFS model is productive, profitable, carbon negative, and climate resilient, offering a scalable pathway for smallholder prosperity. So, the take home message for the study is integrated farming system allows smallholder farms to produce more, earn better, and emit less by functioning as a self regulating agro ecosystem rather than isolated enterprises. This model is not only scientifically sound but also practically viable for smallholders. It offers a clear roadmap for sustainable intensification under climate uncertainty. Here are some reference. Thank you for your kind attention. I shall be happy to take questions. Thank you.
Any online presenter would like to ask a question to Sayani? Anybody?
Any questions, anyone?
Would like to ask a question to Sayani? Anybody? So Sayani, it is a good presentation.
Thank you, sir.
I would like to ask a question.
Yes, sir. Please.
You told this IFS model is will be enhancing a food, nutrition, and livelihood security.
Yes, sir.
I would like to add that even it also is going to give a income security to the
Yes,
farmer.
sir. Yes, sir.
That is one thing. The second one is
Yes,
it
sir.
possible for you to tell, see, the coming to nutritional security
Yes,
or
sir.
nutritional security, the IFS model, how much percentage of nutrition it is contributing to the daily required dietary allowance of a IFS farmer as compared to non-IFS farmer. Do you have any idea? Nutrition it is contributing to the daily. Did you get my question actually?
Hello, sir. Yes, sir. Actually, it is eco, so that's why I am. Sir, actually, this experiment was conducted in Odisha University of Agriculture Technology in the teaching farm. That's why I can't. I could. This experiment was conducted in Odisha teaching farm. That's why I can't know the perfect nutritional percentage. But as the horticulture module was integrated in the system, that's why seasonal nutritional security is confirmed to the farm families. But as the horticulture module was
Okay, actually what I wanted, you can tell that suppose this is the vitamin wise, this means these vitamins are
confirmed to the
the
farm family.
IFS farmer will be getting extra than the non-IFS farmer, or these minerals, micronutrient maybe is iron, zinc or something. These are all things these farmers will be getting say 10 or 20% extra vitamin or extra minerals as compared to non-IFS farmer. Like that, if there is anything is there, you can add. That's what my intention.
Okay, sir. Okay, sir. I will add.
Okay. Because ultimately farmer does not have a money to purchase a fruits or vegetables or pulses in the
Yes,
market.
sir.
This IFS is going to definitely meet out his household requirement of food, nutrition, also even meat or whatever may be. So in that way, definitely IFS farmer will be more healthier than non-IFS farmer. So that we should scientifically prove or give a data so that
Okay, sir.
this, of course you told that it is carbon negative farming. Yeah, it is
Yes,
most
sir.
we wanted a carbon negative farming so that the global warming can be minimized. Okay, good luck. Thank you.
Thank you, sir. Thank you, sir.
Thank you very much, Sayani.
Any other presenter? Vijay Thakur is there. Saida Nabila,
Check.
Banot, Venkateshwarlu.
ଦେଖିବା କିଏ ଅଛନ୍ତି ସେଥିରେ.
Anybody is there? Just I am reading. If anybody is there, he can say yes and he can start presentation. Anoj Kumar, Pooja Goswami,
ପ୍ରେଜେଣ୍ଟର ସ୍କ୍ରିନ୍ଟା ଟିକେ ଦେଖ.
Dorajia, Sunil Nagappa, Rupali,
I think Rupali Gupta is there.
and Sachi. Huh?
Rupali Gupta, is it audible? Are you ready with your presentation?
Yes, ma'am.
Okay, should we go ahead with
Yeah.
your presentation?
Yeah, please proceed, Rupali.
Okay, please welcome Rupali Gupta from Chaudhry Charan Singh, Haryana. Chaudhry Charan Singh University, Haryana, and she is going to present on persistence and eco-toxicological evaluation of butacular and its metabolites in different soil order.
Yes, ma'am. Am I audible?
Yes. Sachin Rajesh Avachar, please be ready with the presentation.
Good morning, all of you. Myself, Rupali Gupta. Today, I'm here to present my thesis topic on persistence and eco-toxicological evaluation of butachlor and its metabolites in different soil orders. So, before proceeding further, I would like to give brief introduction.
So Rupali, 1 minute. Can you put it in a full slide mode? Is it
Yes, sir.
slideshow mode? Please put it. Yeah, now you can go ahead.
So before proceeding further, I would like to give brief introduction, that is butachlor is a selective pre-emergence chloroacetanilide herbicide used to control broadleaf and grassy weeds in rice. Photo and microbial degradation of butachlor leads to the formation of different metabolites. These metabolites have different physicochemical properties which may influence their environmental persistence and toxicity risk to non-target organisms and surrounding environment. Different type of soils may differently affect the ultimate fate of these metabolites and their movement in surrounding environment. The environmental behavior of different metabolites and their toxicity effects on non-target organisms are rarely reported. So here is the proposed metabolic pathway of butachlor. Butachlor, when they are applied or when they are applied in soil, they get degraded due to microorganisms, or there leads to a photo degradation, which leads to the formation of other transformation products, such as when the debutoxylation occurs, they forms compound which are named M3, that is 2-chloro and 2,6-dimethylphenyl and methyl acetamide. And further, due to demethylation, they leads to the formation of 2-chloro and 2,6-diethylphenylacetamide. And due to further dechlorination, these form the compounds M2, which is named as 2,6-diethyl and acetanilide. And further deacetylation causes leads to the formation of M5, that is 2,6-diethyl-aniline. And for here, due to the photodegradation, dechlorination occurs of butachlor and forms the compound N-butoxymethyl and 2,6-diethyl phenylacetamide. And due to this, the last product formed that due to the deamination, that is M4 2,6-diethylphenol. So here is my experimental design. I took 4 soil orders, that were aridisol, antisol, inseptisol, and alfisols from Haryana. And the herbicides which were studied, that were butachlor and their its metabolites. Application rate was 1,500 gram per hectare for T1 dose and 3,000 gram per hectare for T2 dose. And the third treatment that was taken was untreated control. So the three replications were taken, and sampling days were 0th day to 90th days after application. So here is the first result of my findings, that at T1 dose, that is a single dose which were taken were 1,500 gram per hectare, but the active ingredient which are used of butachlor are 50%. That's why the T1 dose that was calculated is the 750 gram active ingredient per hectare. They were applied in the soil and their metabolite as well. Butachlor M3, M4, and M5 were studied. At T1 dose in alfisol, the initial residues were found to be 0.155, and due to there the percentage dissipation occurs up to the 30 day for butachlor, and M3 persists for shorter period of time in the soil in alfisol, and they persist for up to 7 days, and after 7 days there is a below detection limit that was found. For M4, here we can clearly see that M4 persists for longer period of time, just like butachlor, up to the 30 day after application, and M4 persists for 15 days. Here are the half-life of the butachlor, that is 10.4 days, and for M3 it is 2.19, for M4 it is 17.64, and for M5 it is 8.9. Here are the graphs. The first graph represents the dissipation behavior of butachlor and its metabolite in alphy soil. From clearly we can see that M4 and butachlor persist for longer period of time, that is 30 days after application, and M3 persist for shorter period of time. Next is when they are applied at double dose, their T half is 12.7 days for butachlor, and for M3 it is 2.52 day, and for M4 it is 19.2 day, and for M5 it is 9.7 day. Here at T2 dose, the result is same, but M4 persists for longer period of time. That is 45th day after application, and but butachlor and M5 persist for 38th day. Here also M3 persists for shorter period of time. Next is when the butachlor and their metabolites were applied in inseptisol soil order. Here the results are quite different from the alfisol, because inseptisol has low organic matter, so the persistence of butachlor as well as their metabolites are comparatively less. They dissipate at a faster rate than alfisol. So the T half of butachlor is 8.8, and for M3 it is 1.8. For M4 it is 13.4 day, and for M5 it is 7 day. So here is the graphical representation from where it is clearly visible that the butachlor and M4 persist for 30 day after application, but M5 persists for 50 day after application. Here also M3 dissipates at a faster rate, and they persist for 7 day after application. So here is the T2 dose for inceptisol when the doses are double. There the persistence also the metabolites and butachlor persist for longer period of time in inceptisol, and here are the half lives was is 9.9 days for butachlor, 2.3 day for M3, and it is 15 day for M4, and then it is 8.3 day for M5. Here are the graphical representation. As we can clearly see that dissipation behavior of butachlor and its metabolite in inceptisol at T2 dose, where we can clearly see that M4 and butachlor as well as M5 persist for longer period of time, and they dissipate slowly as compared to M3 and persist for 30 days after application. But M3 dissipates at a faster rate as compared to other metabolites as well as parent compound. From here, the second graph also represents the semi-logarithmic graph showing first order kinetics of butachlor and its metabolite in inceptisol at T2. And the third order, that is antisol at T1 dose, the half-life of butachlor is 6.1 day, and for M3 it is 1.4 day, for M4 it is 11.4 day, and for M5 it is 5.4 day. From here it is also clearly visible that in antisol they persist for shorter period of time as compared to other soil orders. Here are the graphical representations for butachlor and its metabolite. Here also it is clearly visible that M4 persists for longer period of time as compared to the parent compound, as well as M3 dissipates at the faster rate. In when the dose is applied at when the double dose is applied, the T half of the butachlor is 7.2, and for M3 it is 1.8, for M5 it is 13.9, and for M5 it is 6.7. So here are the graphical representation. Here it also represent that M4 persists at double dose also. M4 persists for longer period of time and dissipates very slowly, but M3 dissipate at a faster rate. When the now here is the eridisol, the next fourth soil order which was taken for butachlor. Half life is at T1 dose. When the single dose is applied at T1 dose, the half life is 4.8 day, and for M3 it is one first day, and at T1 dose for M4 it is 8.1 day, and for M5 it is 3.8 day. Next, here is the graphical representation. Here also it is concluded that M4 persists for longer period of time, but M3 persists for shorter period of time and dissipate at a faster rate. So at double dose, when the dose were applied, T half of the butachlor is 5.5 day, and for M3 it is 1.2 day. For M4 it is 9.4 day, and for M5 it is 4.4 day. So from the graph, it is also it is visible that here also M4 persists for longer period of time, and M3 dissipates at a faster rate. So here are the comparison of half-lives at all the soil orders which were taken at T1 and T2 dose. At T1 dose, we can clearly see that green color represents butachlor, red color represents M3, and purple color represents M4, and that blue color represents M5. From here, it is clearly visible that there are the four soils which were taken under study, and the half-life of the M4 is more in all the soil orders as compared to the parent compound as well as the metabolites, and the half-life of M3 is very less in all the soil orders. Here also the comparison can be made in the soil orders, as in alphy soils, the metabolites as well as the parent compound persist for longer period of time as compared to inceptisol, followed by antisol and eridisol. The reason behind is because the alphysol has higher organic matter content. So this is the reason the parent compound as well as the metabolites persist for longer period of time, and the similar results were found at the double dose. And when the double dose were applied, their half lives they persist for longer period of time, and the order was same. In alphysol the persistence were higher, and then inceptisol followed by antisol and then eridisol. Here results are also same, that M4 persist for longer period of time in the soil for all the soil orders, and M3 participates at the highest rate in all the type of soil orders. So the next slide represents the comparison of below detection limit of the metabolites as well as the parent compound. From here, they are the graphical comparisons. From here, it is visible that at T1 dose in all the soil orders, the butachlor and the metabolite 4 they persist for longer period of time as compared to other metabolites. And in alfisol, both butachlor as well as the other as well as M4 and parent compound persist for longer period of time, and their below detection limit was found to be 45th day. And same for inceptisol, but in antisol, the persistence of butachlor changes as they persist for shorter period of time as compared to M4, and in M3, as always, M3 persists for very short period of time, and the below detection limit is very less. And same goes with double dose. So here were the toxicity evaluation which were evaluated at various non-target organisms, that was in algae, earthworm, honeybee, fish, birds, and rat. From here, it is concluded that in alfisol, algae, earthworm, and fish were at high risk up to the 30-day, and birds and rats were at a low risk from the initial application of the metabolites in alfisol. In the same results were found in metabolites also at up to the as M3 persists for up to 7 days. So their toxicity in algae, earthworm, and fish is very high at up to the 7 day, but in birds and rat their toxicity is very less. In insectisol, the order was as earthworm, algae, and fish were at high risk up to the 30-day, and birds and rat were at low risk as their initial from the initial dose their risk was low.
So kindly come to the conclusion. Your time is over, no? थोड़ा सा जल्दी से करिए, conclusion
sir.
पे आइए.
Same trend was followed in antisol and as well as aridisol. So the conclusion is that butachlor and M4 persist for longer period of time in soil in all soil orders, and M3 persist for shorter period of time, and the toxicity risk is low. Thank you.
Any question from the online participant to Miss Rupali? So Rupali, what is the relationship between soil organic carbon and the toxicity of different weedicides?
So when the organic matter is high, due to the high organic matter, butachlor, as because they are also polar compounds, so they persist for longer period of time in soil as compared to the soil order which have less organic matter. So this is the reason alfisol has more organic matter, so the compounds persist for longer period of time as compared to aridisol, which has low organic matter content.
So you want to say that if soil is having more organic carbon content, we should avoid herbicide application, or
yes sir, we can, from here we can conclude that those soil orders which have a high organic matter content, butachlor should not be applied, or as well as their metabolites. In fact, some of the metabolites had higher toxicity than butachlor.
so you have not studied the effect of these weedicides on the soil microbes?
No sir, no, not on microbes.
Okay.
So I have studied their sorption-desorption behavior as well, and their and their effect on enzymatic activities in soil present, like the hydrogenase beta glucosidase and urease, alkaline phosphatase.
So which instrument you have used for measuring all these things?
So HPLC.
HPLC.
Yes sir.
Okay. So it's really very interesting study that different soil is behaving differently for the different weedicides. Am I correct?
Yes sir.
Based on the soil type, we should. I think there is some issue with her. Okay. Yeah. Hello.
Yes, sir.
Means different soil types also play an important role in selection of weedicides, huh?
Yes, sir. Because their soil texture is different. They have a different, as like alfisol has more clay content, clay percentage, and aridisol has more sandy texture.
Okay, thank you. It's very nice presentation. Thank you.
Thank you, sir.
Okay. Who is next?
Thank you very much, Rupali. Moving on, over to Sachin Rajesh Avachar from Suwards, Prayagraj, Uttar Pradesh.
Yes, ma'am. Good afternoon, all of you.
Very good afternoon. He's going to
Yeah,
present.
please go ahead. You are audible and you can share your PPT. Yeah.
Yes, followed by Manasvi Dora. Sachin is going to present.
Okay,
ma'am.
Genetic divergence analysis of maize hybrids for morphological and quantitative traits under Prayagraj agroclimatic conditions. Sachin, over to you.
First of all, good afternoon, all of you. This seminar present my research work, which is genetic divergence analysis of maize hybrid for morphological and quantitative traits under Prayagraj agroclimatic conditions. The study was carried out in the Department of Genetics and Plant Breeding, Shouts, Prayagraj. The the presentation is organized into introduction, objective, and justification. Maize is one of the most important cereal crop worldwide due to high yield potential. It is known as queen of cereals also because it is used in wide adaptability and multiple uses, just like it is used as food for humans, feed for livestock, and raw material for industries like starch, ethanol, and biofuel. Maize originated in Mexico and Central America, belonging to the Poaceae family. It is high adaptable crop grown for tropical and temperate regions. Due to the genetic plasticity, maize perform well under diverse environmental conditions. Where if you talk about the area and production productivity in global and India, globally maize is cultivated in more than 170 countries. It has the highest production among cereals and contributes significantly to food security. India is one of the most leading maize producing countries with cultivation in both Kharif and Rabi season, and also in spring season also, which is called Z1. And in as we talk area production and productivity of Uttar Pradesh, in Uttar Pradesh maize is gaining importance due to the rising demand from poultry and starch industry. The state has considerable scope for increasing maize productivity. Genetic improvement through hybrid evolution can help to achieve the higher yields. This study has some objectives to just like to estimate genetic variability, heritability, and genetic advance for among maize hybrids, to study genetic divergence using Malnobis, that is called D-square analysis, and to identify superior hybrid for future breeding programs. The study shows which material used in that. The present investigation was conducted during Kharif 2024 at Shorts, Prayagraj. The experimental was carried out under the field condition following recommended agronomic practices. Sorry. The total number of hybrids used in 26 hybrids were used. The experimental was laid out by appropriate experimental design with replications. The experimental material was provided by CIMET from Hyderabad. Here is a methodology. Days to 50% flowering and days to maturity was recorded as assessed crop duration. Plant height and ear height was measured in the growth behavior. Lodging and number of ears per plant were recorded. These are 15 number of observation were recorded. On the analysis, the data were analyzed by analyzing the variance of test of significance and the genetic parameters such as GCV, PCV, heritability, and genetic advance were estimated. So we can come up to a result. The significant difference were observed among these maize genotypes of most traits. The result confirm present sufficient genetic variability. The ANOVA relevant highly significant variation among these genotypes. The indicated reliability of the experimental and scope of horse selection. And for the genetic parameters, the phenotypic coefficient of variation was higher than genotypic coefficient for most of the traits. High heritability along with uh, high genetic advance were observed for important yield traits, indicating effectiveness for selection. And we can come up to cluster analysis. The 26 maize genotypes were grouped into 3 clusters. The clustering indicates the presence of genetic diversity among hybrids. The dendrogram shows genetic relationship among maize genotypes. Genotypes in the different clusters are genetically diverse. And we can talk about intra and intercluster distance. High intercluster distance suggests wider genetic divergence, such genotypes are suitable for hybrids for obtain better recommendation. And we can come to conclude that VH171310 and JDH 221149 were indicate higher superior genotypes. Trade likes number of years per plant, field weight, and grain weight were important. These traits can be effectively used in maize improvement program under Prayagraj region. These are observations recorded in actual field condition. These are pictures, tassel emergence, silk emergence, plant height, ear height, number of kernels per rows, and number of kernels per rows, different shape, size of color of ears, and ear length by taking in centimeter. And last one is grain weight. Okay, thank you.
Okay, thank you. Any question from online presenter to Mr. What is his name?
Sachin.
Sachin. Okay. Any question to Sachin? Sachin, you told me that number of years per plant, right?
Yes, sir.
So what was the variation you got it from different genotype? What is the lowest number of years per plant and highest?
In a maximum number of years, there are observed 1 years per plant. And if you talk about the number of year maximum, there are 3 number of years, which is a maximum in a number.
In a single plant, 3 years also observed, some jump plus and
Ha,
something.
3 years also observed, and most or commonly 1 or 2 observed.
So means if 3 years are per plant, then yield is more or what? Or quality yield is less, total yield.
Okay. If we talk about the micronutrient, अगर दो जो हमारा ईयर से वो अच्छी तरह फिल हो चुका है, तो तीसरा नंबर का ईयर है जो उस पे हमको क्या हुआ है, कम माइक्रोन्यूट्रिएंट मिला है उसको, तो उसका थोड़ा डेवलपमेंट कम रहेगा, तो उससे यील्ड पे मैक्सिमम मतलब 2 का ये एवरेज ऐसा मीन परफॉर्मेंस हम देख सकते हैं.
You try to speak in English only because I don't know much.
Oh, okay.
That is
Uh,
it's fine.
if
Huh?
there is a three number of years, then the number of the third person third year is in a yield in low in weight also, because two has developed very well, but third one is not getting any some kind of nutrient only, so the average will be 2 years.
So in general, 2 years per plant will be giving higher yield than the
Higher
3.
yield. Yes, sir.
Okay. Okay, good. Thank you.
Okay,
Yeah,
thank
please. Yeah, sir, want to ask
सचिन
our question.
सचिन
हाँ
दो चीज़ें आपसे पूछना चाहता हूँ। पहली चीज़ ये
जी
कि
सर।
जो हाइब्रिड्स आपने इवैल्यूएट किए, whether
हाँ
you
जी सर।
whether you have taken diseases, incidence of disease and pest as a parameter or not.
यस सर,
आपने दिखाया नहीं
पैरामीटर्स
इसमें।
नहीं रखा है, पर ऑब्जर्वेशन में आया है कि उसमें अटैक हुआ है हेलिकोवर्पा आर्मीजेरिका।
नहीं, वो तो होगा ही है जो अगर मेज लगाया
हाँ
होगा, लेकिन कंपैरिजन व्हेन यू हैव
You have not
compared
included in the analysis also,
whether you
no?
have included as a parameter in the analysis that you are presenting here, yes or no?
हाँ यस सर।
नहीं, आपने इंक्लूड नहीं किया शायद।
जो 15 parameter
नहीं,
है
इसमें इंक्लूड नहीं किया है सर, वो बस
ठीक है,
ऑब्जर्वेशन में आया है
okay, no,
ग्राउंड
we are
पे।
concerned only what you are presenting here. First
हाँ
thing.
हाँ हाँ
Second thing, you have evaluated all hybrids.
हाँ यस सर।
All hybrids. So
All hybrid.
I am loss of knowledge whether hybrids can be used as parental lines. Yes
सर
or no?
अभी नहीं यूज कर सकते उसको,
No, no, no,
इनब्रीडिंग
no,
करेंगे
no,
हम
no,
उसको
no, no,
सेल
no.
में
आप
करेंगे।
मुझे
हाँ
सीधा सीधा आंसर दीजिए, स्ट्रेट आंसर. Whether
हाँ
hybrids can be used as parental lines or not.
नो सर।
No. आपका कंक्लूजन में आपने कहा है कि these hybrids can be used as parental lines. तो ये आधारभूत गलतियां मुझे ऐसा लग रहा है कि शायद हैं, तो आप थोड़ा उसको आगे सुधारिए, उससे शायद आप
ओके सर, आई विल इम्प्रूव सर।
इन दो चीज़ों को आप करिए, क्योंकि ये मतलब बेसिक इसको कह सकते हैं कि मिस्टेक्स है, ठीक है?
For the breeding program you can use, but not as a parent.
You cannot
ठीक है?
use it as a parent, ठीक है?
यस सर। ओके
You are
Okay.
a breeder
Thank
ना
you.
भैया, आपको
Okay,
तो
okay.
मालूम होना चाहिए.
All the best, all the best.
जी सर, आई विल इम्प्रूव सर।
Okay. Then I think Sunil, Manasvi. Okay. So after Manasvi, maybe Sunil, right? Yeah, Manasvi, please go ahead.
Yes, sir.
So Manasvi, what is her topic? Where is
Good. 1 minute, sir. 1 minute.
आप ना एक सिस्टम यूज करिए, दो सिस्टम यूज मत करिए, ओनली यूज वन सिस्टम। हाँ,
you are not
Am I
audible.
audible, sir?
Yeah. Now you are audible, I think. Go ahead.
Okay. Good afternoon, respected chairman and co-chairman and all of the participants. Today I will be presenting a review on the topic of nanopesticides, a modern solution for sustainable plant protection and precision management. As we all know, there are the various challenges in agriculture. There is a crop losses up to 20 to 40%, and more than $220 billion dollars annual damage is there, and that is caused by the chemical pesticides concerns, and environmental impact is also is there when we use broad spectrum insecticide and all that. So there is also affect the non-target organism, and also affect the environment, soil, and that leads to the pollution. So nanotechnology that will solute us that we can use as the nano encapsulation, that is a controlled release in the we now there is available of nanoparticle enhanced pesticides. Also nano formulation is there, RNA technology is there. There is a polymer capsule we can use as a metal nanoparticles for the pesticides, and that leads to the smart pest monitoring. These are the various formulation available in the for the nanopesticides, that are the nano emulsion, nano suspension, polymer based, lipid based, clay based, metal based. There are the various formulation that we can also make for the insect and pesticide making. There is the various benefits of nanopesticide that leads to the target action, and also that reduce the chemical use, also the environmental friendly and enhance the efficacy and lower toxicity to the non-target organism. So it will be safe, and that is lead us to the sustainable crop protection. And there is a various other application in agriculture also there. We can use as a soil remediation, water management, crop management, and post-harvest solution. This graph shows that the global use of pesticide in agriculture has increased from approximately 2.3 million tons active ingredients in early 19s to more than 4 million tons in 2016. So this graph shows that there is the increasing of insecticide and pesticide use into this, and this chart shows the total agriculture use of pesticide on the different continents. So this shows that Asia uses the highest pesticide use as compared to other continents. So that is the very higher issue in this in our continent for pesticide management. So what is the solution? Solution is we can use this as a nano pesticide as compared to the other pesticides. So how can we make the nano pesticides and green synthesis of the pesticides? So first of all, we can make for the making this we can take to the green plant and extract the plant powder. Then they mix the plant molecules with boiling at a 60-degree temperature plus water, and then filtrate them. Also at that time we can add the nanoparticles into this, and then nanoparticles coated with the green our plant powder. So that is the green nanoparticles synthesized from this, and this after synthesis of nanoparticles, we can use as the application of the foliar and soil also. In the foliar application, we can use it as a pesticide, nematicide, bactericide, and also we can enhance the soil reaction, and also we can use it as a fertilizer also. And we can use it a pathogen and pest management in plant. This is the there is various biopesticide also is there. We can encapsulation of biopesticide within the nanoparticle. Biocontrol agents like BT and neem oil encapsulated within the natural matrix based on the nanoparticle offer increased stability and efficacy for the pest control. We can also Bacillus thuringiensis subspecies israelensis and the silver nanoparticle is enhance the pest control for the enhance the pest control, and encapsulating the biopesticide within the natural matrix based nanoparticle offer increased stability and efficacy for pest control. This is there are the various review that I collected from different sources. There is the mechanism of action of the nano pesticides. So there is the increased solubility, also targeted delivery, also that enhance the penetration and control release. Also it is the drift leaching prevention and synergistic and non-toxic breakdown, that adjuvants improve efficacy and nanoparticles break down into the harmless byproducts after active ingredient release. And then regulatory advancement is there. So development of standardized safety and risk assessment protocols facilitate the large scale production and farmer level adoption. There is the various nanoparticles we can use as a silver nanoparticles, copper, zinc, chitosan, and polymeric. Mostly used as a chitosan nanoparticles. Silver nanoparticles that penetrates microbial cells, cell membrane, and internal component, affecting the crucial cellular operation and resulting in cell death. And chitosan nanoparticles increasing the protective genes like different antioxidant enzymes, as well as the elevation of the level of the phenols. So silver this figure shows the how silver nanoparticles attack the bacteria by releasing silver ion that damage the bacterial cells and stop the infection. So we can use this as a bactericide, and also we can use to treat the disease, bacterial disease also. Then chitosan nanoparticles attacking the cuticle of the insect, so that leads to the attacking the cell membrane and oxidative stress reduction, and that leads to the death of the insect. Then the chitosan nanoparticles that also attack on the nematode also. So this chitosan nanoparticle attacking the eggshell of the nematode, and then the eggshell of nematode break down to the chitinase due to the chitinase activity. Also, this shows the chitosan nanoparticles delivering nematicides to the root zone. So plant chitosan nanoparticle carrying out the direct attack on the nematode, and that leads to the we can control of the nematode also with nanoparticles. There is a various nanoparticle enhanced insecticide is there. So ivermectin is a bactericidal and antiparasitic agent targeting the nematodes and arthropods. So there is a super carriers to load and control ivermectin release. So how can we release the ivermectin? So we can use it as a nanosilica, nanocapsule, nanosphere, nanogel, micelle, and liposome also. These nanomaterials have been used to encapsulate the insecticide in various forms, including nanocapsules, nanosphere, nanogel, micelles, and liposomes. So these encapsulated formulation have demonstrated and enhanced the insecticidal effect and longer lasting residual activity as compared to their commercial counterparts. So some challenges are also is there, not completely there is not completely perfect. So some challenges are there, regulatory and safety hurdles, economic and industrial scaling, environmental impact, and also that economic and industrial scaling challenges are there, social and collaborative barriers like addressing fear of unknown that regarding nanotechnology in food production through better communication, and social and there is various barrier is there, and this is the future thrust of this nano pesticides. So this nano pesticide targeted and controlled delivery, then we can use as a sustainable pest and disease management. Also we can use as a precision agriculture and smart farming, because we can install the nano sensor and that allow the real-time monitoring of the crop and soil health. We can use as the enhanced crop productivity and stress tolerance, and environmental and human health safety and regulatory advancement and commercialization. At last, we can conclude that the conventional chemical pesticides suffer from the low efficacy, with over 90% of the active ingredient lost of the environment through leaching, evaporation, and runoff. So this review explores the emergence of nanopesticide as a transformative solution for the sustainable agriculture. Also classify various formulations. Also classify the various nano formulations is there. So evaluate their role and reducing the chemical footprint into soil and water. So this paper critically evaluate the synthesis of the green nanoparticles and their role in improving the photostability and target specificity of the pest control agents. So this discussion highlights the reduction in chemical footprint and mitigation of the groundwater contamination. And by integrating nanotechnology with IPM strategy, nanopesticide offer the higher efficacy, low dosage pathway to resilient agriculture. So this review conclude by addressing the current regulatory hurdles, the toxicity assessment required to ensure the safety of non-target organism and sustainable agriculture ecosystem. Thank you, sir. These are my references.
Any questions from online participant to Manasvi? Co-chairman sir, would you like to put any question?
मनस्वी जी अभी बताइए कि इज देयर एनी एग्जांपल ऑफ मैनेजमेंट ऑफ वायरल डिजीजेस इन प्लांट्स यूजिंग नैनो पार्टिकल्स
नैनोपार्टिकल
वायरस डिजीज
हाँ सर, हम वायरल डिसीज का तो हम एज अ वेक्टर हम कंट्रोल करके उसका
नहीं
मैनेजमेंट तो
डायरेक्टली targeting the viruses in plants. Is there any technology is there? Nanoparticle technology. Are you aware?
सर वो तो मुझे आइडिया नहीं है सर।
Okay, ठीक है. That is first question. Second question is that have you come across any clay particle nanotechnology? आपने लिस्ट किया है प्रेजेंटेशन में clay
हाँ
particle. Are you aware of any example? Successful example? नहीं
सर
तो कोई बात नहीं so you have already enlisted in your slide regarding RNAi technology which is
हाँ
actually
सर
can be used for viral disease
हाँ
आपके स्लाइड में ही है और इवन क्ले पार्टिकल का भी आपके स्लाइड में ही है और ये
हाँ
बहुत
हाँ
ही सक्सेसफुली इसको यूज़ किया गया है इन avocado in Australia by one Indian scientist. You can go through that. Thank you.
Okay, Manashwi, it is only a review, right?
यस सर
So when you present a review, na, you
हाँ
should, what type of analysis you have used? Some meta analysis are there. Have you used that one? Then how many paper you have shortlisted? Out of that, so many nano your clay based that one. How many manuscripts are dealing with all those things? Those type of uh, those type of information you should include.
ओके सर
Okay, in your presentation, whenever you do the review and review paper review presentation, you should come out with a meta-analysis totally whether thousands of literatures, how many it
ओके
was related to on pesticide, then out of the different type of pesticides. So like that you should come out with that one. Okay,
ओके सर
okay.
आई विल
Otherwise,
इम्प्रूव
yeah, okay, that's fine. Okay, thank you. Good. So next, who is next? Sunila?
Yes, sir. Present, sir. Sunil.
Yeah, please go ahead, Sunil.
Okay, sir.
So, Sunil Nagappa, he will be presenting on the effect of natural vitamin E, C, and choline and herbal liver supplement on serum biochemical parameters in commercial broilers. Okay. I think, yeah, she can unshare.
Hello.
Her name, 1 minute. Manashwi can unshare. Yeah, it's fine now, yeah. Sunil, your slide is visible, Sunil. You can go ahead.
Okay, sir. Thank you, sir. So good afternoon, respected chairpersons and judges, delegates. Myself, Sunil Kodler, MSc scholar from Department of Poultry Science, Veterinary College, Hebbal, Bidar. So today I will be presenting my thesis research on the effect of natural vitamin E, vitamin C, and choline and herbal liver supplements on serum biochemical parameters in commercial broilers. So come to the introduction part. In broiler production, the feed accounts for nearly 70 to 75% of the cost. So improving feed efficiency and bird health using safe additives is very much essential. Due to restrictions for antibiotics and for residue-free meat or natural residue-free meat, natural feed additives like herbal products and vitamin supplements are gaining very important. Therefore, this study was conducted to evaluate the effect of natural vitamins and herbal liver supplements on serum biochemical parameters and other growth parameters also. But here I am presenting just biochemical parameters which come the good results. So results part just
Your
some
slides are not
poultry
moving.
support is
Sunil, your slides are not moving. Yeah, now it's moving. Yeah, go ahead. You are in My Poultry
so
Needs.
why poultry these vitamins or these nutritional. So basically, modern broilers grow rapidly are highly susceptible for metabolic stress and oxidative stress, other liver dysfunction, especially under heat stress conditions. Such stress affects the nutrition utilization and also. So nutritional intervention is required to support and antioxidant status and the metabolic efficiency too. These are the main. Hello.
Yes, Sunil, your PPT disconnected. Can you share one more time?
There's someone sharing is some other.
Okay. Okay, go ahead.
Now it's visible, sir.
Yeah, visible.
Sir. So coming to the main the physiological roles of these vitamins. For example, vitamin C, it's a very good antioxidant and stress resistant and immune modulation. And coming to vitamin E, it is also a very good antioxidant, protects cell membranes and immune booster. And coming to choline and liver supplements, these are obviously choline is for fat metabolism and liver function and neurotransmission. It is the main role in poultry or any other animals too. So why natural or herbal? Why we used or why we are interested in the natural or herbal products? The first thing is these products are more stable due to natural matrix and protective phytochemicals present in these products, and a superior bioavailability and antioxidant capacity compared to the synthetic ones. So, and higher retention in plasma, muscles, and liver, which is beneficial for shelf life and nutritional value. And coming to the economical part, it is on par with synthetic too. It's not more costlier or less, which is on par with the synthetic. So economical also, less dose. And compared to synthetic, it's we used very less dosage and more efficacious compared to synthetic. And obviously eco-friendly, residue-free, and sustainable poultry production. So consumer needs are also fulfilled. The main objectives of our studies to study the effect of obviously natural vitamin E, C, choline, and herbal leaf supplement on serum biochemical parameters. The parameters studied here: SGPT, SGOT, serum cholesterol, and serum triglyceride. This is my experimental design. Here we used 150 broiler chicks. 150 broiler chicks were divided into control and treatment groups receiving a natural vitamin E, vitamin C, choline, and herbal liver supplements. With the T1, it is basal diet formed prepared as BAS standards 2007, and T2 is basal diet plus 0.1% natural vitamin E. T3 is basal diet plus 0.1% natural vitamin C, and T4 is natural choline, which is it is also 0.1% we added. In T5 also 0.1% herbal liver supplement we added extra. In basal diet, that is control, we didn't use any of these additives. So directly coming to the results and discussions, we got a very good results in all among all the supplements. First, we got a significant decrease in serum cholesterol and serum triglyceride in group supplemented with natural vitamin E compared to the control group. There was significant decrease in SGOT, serum cholesterol, and serum triglyceride in the supplemented with the natural vitamin C compared to control group. And SGPT, serum cholesterol, and serum triglycerides is significantly decreased in natural choline. And SGPT, SGOT, and serum cholesterol in herbal liver supplement treated group compared to natural. In short, this is the in short that in natural vitamin E, we got decreased cholesterol and triglyceride. In vitamin C, other than SGPT, we got all the other three parameters with significant decrease, and choline also. The herbal liver supplement, SGPT, SGOT, and serum cholesterol. My results are with per and present studies are Jadhav and Co. and as well and Co. who who show that serum cholesterol and serum triglyceride values shown a significant difference, supplemented group and control group. And also the results of the present study are in also agreement with Dhumal and co, who observed a significant decreased values for SGOT patent herbal liver supplement, herbal liver stem supplemented groups. And also Dhumal and co, who observed the herbal choline treatment group shows a significant improvement in serum SGPT activities. The main important thing that the result is natural vitamin C treated group showed the significant lower serum cholesterol and levels due to the presence of which tannins, the travel and body fats, and herbs such as nilotica and longa. The present product, the choline, the main ingredient, are rich in and curcumin. These are basic. Hello, sir.
Yeah, please go ahead. Sunil, you go ahead. We could not hear you.
These herbal liver supplements act as a hepatoprotectant. Sir, I'm not audible, sir.
You are audible. Go ahead, please.
Okay, sir. So the herbal liver supplements act as a hepatoprotective and liver tonics, prevents the liver damage and reduce the leakage of liver enzymes. So that's why we got the liver enzymes very significantly reduced compared to the that is non-supplemented control group. So coming to the conclusion, natural feed additives significantly influence the serum biochemistry, and herbal liver supplements showed the strongest hepatoprotective effect. Also, natural choline improved the lipid metabolism. So coming to the practical implications, improves the liver health and metabolic efficiency, reduce dependence on antibiotics, supports sustainable poultry productions. Thank you, sir.
Thank you. Any question from online participant to Mr. Sunil? Sunil,
Sir.
hear me?
Yes, sir. Yes, sir.
Yeah. So use of these natural sources, can we call it as a organic broiler cultivation if I grow a broiler using this one?
No, sir. By only using these herbal products or natural, we can't conclude it as organic, sir. Other practical management is also very important, sir, as to conclude as an organic product.
But this is a organic. If I use this natural, this one, it is allowed for organic.
Yes, yes, sir. Yes, sir. You can. It is obviously we can use as a part of the organic production, sir. Obviously, sir. If we also manage other managemental practices like non-using any other antibiotics or synthetic products, and by using these additives like this and other additives, herbal products, we can obviously conclude it as organic products, sir.
Okay, I got it. So in your materials and methods, you have not your treatments are there, but what happened to that inorganic vitamin E or vitamin C? It is not included in your materials and method. Then how you compared? You are telling you have compared a natural vitamin E with that I think inorganic vitamin E, but in your treatment data
No, sir.
only 5.
No, sir. We didn't compare to synthetic, sir. Actually, we just compared with the basal diet, which is not supplemented with vitamins, sir. That basal diet, it is not supplemented with these vitamins, sir. Vitamin E, C, choline, and liver supplement.
Fine, but one treatment then you should have included use of this artificial means what synthetic vitamin E and C and all as one treatment by
Yes, sir.
Okay, then only you are telling that natural vitamin E, C supplements are better than synthetic. But when you have not included them as a one of the control, how can you say like that? That is one
Yes.
thing. Second one is you have evaluated separately what is the effect of vitamin E, C, choline like that. What will happen if I combine all the things as a one treatment and feed it to the, because definitely vitamin E also required, C also required, choline also is required.
Yes, sir.
I feel it is very poorly designed methodology. That's what I don't know. I am not a expert of veterinarian. As a
Sir,
layman.
sir, yes, sir, yes, sir.
Okay, anyway, thank you. No issue.
Thank you.
So, who is there? I think Bidisha. Pooja. Pooja, I think she is, I think presented or not?
Yes, sir.
Okay. Pooja, can you hear us? You can present.
Yes, sir.
Yeah,
Yes,
you
sir.
can present.
Okay, sir.
Vicky. Who is Vicky? Can you tell who is Vicky? Can you name yourself? Hello? Vicky, what is your full name? Can you? Vicky, can you hear us? Pooja also disconnected. Huh? She was there. Yeah.
Vicky, if you are there, please name yourself. There is no name as Vicky. We should know your proper name. Another is Bidisha Sarma.
There are maybe
Are you one
just
participant?
online person, maybe present.
Are you going to present? Vicky, am I audible? Okay, let's move with the
Banot also is going to present?
Yes, yes. His name is there.
Okay, Banot, can you share your slide?
Sir, good afternoon.
Good afternoon now.
Only
Banot
1
from
minute, just wait, Banot. I think Pooja is ready. Are you ready, Pooja? If Pooja is ready, after Pooja, you can present Banot. Okay.
Hello, sir. Am I audible?
Yes,
Yeah,
yes.
you are audible. Share your slides.
Is my screen visible, sir?
Yeah.
Okay. Good afternoon, everyone. I'm Pooja Goswami, PhD student from GB Pant University of Agriculture and Technology. I'm here to present my results on topic from participation to prosperity, impact of National Rural Livelihood Mission on livelihood security of rural women. Going to the introduction, the Government of India has launched the scheme National Rural Livelihood Mission in June 2011. It is to implement the new strategy of poverty alleviation woven around community-based organization. Its main focus is on universal inclusion of poor that are identified through participatory process and to extend support to access credit and services for the sustainable livelihood and improved quality of life. The program's end goal is the poverty reduction. The program mainly attempts to do this by
Sorry for the
creating.
interruption. Pooja, can you hear me? Can you make it in landscape mode? Please share the full slide. Yes, please go for it.
Okay. Now the program attends this goal by creating SHGs that are effective, building their capacity to self-manage and federating them, enhancing women access to formal credit and promoting savings, promoting livelihoods with a particular focus on enterprises and skilled employment, converging with various existing schemes and institutions for efficient delivery of social, economic support services for the poor. Now, with this program, the SHGs are the key pillar. These SHGs are federated at the village, gram panchayat, cluster, and block level. The institution provides services, mainly savings, credit, livelihood support, to the SHG member that help them strengthen and sustain their livelihood. Till June 2025, 10.05 crore women from rural households have been mobilized into more than 90.9 lakh self-help groups. Now, the concept of livelihood, according to Chambers and Conway, the livelihood refers to the capacities, assets, activities, and coping strategies that people use to sustain life and well-being. Livelihood security mainly means adequate and sustainable access to income and resources, mainly to meet basic needs such as food, shelter, education, health, and social participation. It also recognizes vulnerability to shocks such as economic, environmental, health, and highlights the need for resilience building measures. Now, the objective of the study was to study the impact of National Rural Livelihood Mission on livelihood security of rural women. The universe of study was selected as Uttarakhand. In Uttarakhand, two districts, that is Udham Singh Nagar and Pauri Garhwal, selected purposively on the maximum intensive who have the maximum number of SHGs under this scheme. Similarly, two blocks from each districts were selected, that is Khatima, Sitarganj from Udham Singh Nagar and Dugat Thalisain from Pauri Garhwal. Similarly, two villages were selected from these blocks and two SHGs were alternatively selected from these villages. A total of 160, and all the members of the SHGs were selected from those SHG. A total number of SHG members were 162. Now, to compare this group, a control group was selected of non-SHG members of 100 respondents. A total rural women respondent were 262. Now, the data was collected through a pre-tested interview schedule. Now, livelihood security was mainly organized as the adequate access to income and other resources to meet basic needs, and it was measured with the help of index developed by Gotham in 2021. The reliability of the livelihood security index was also assessed using Cronbach alpha. The overall reliability coefficient obtained was 0.82, indicating a good internal consistency. And the indicators for measuring this livelihood security was food security, economic security, health security, educational security, social security, institutional security, and infrastructural security. Like now coming to the result and discussion, propensity score matching was used to analyze the impact of this this scheme on livelihood security of the women. After that, matching of the sample is formed. The final impact assessment was carried out on the 224 observation. The magic process ensured that the both treatment and beneficiary group, that is control and beneficiary group, were balanced on the key socioeconomic and demographic characteristic before estimating the impact. The table also represents the same. This demonstrates that the PSM approach achieved good covariate balance, allowing for unbiased estimation for the program impact. Now coming to the average treatment effect on the livelihood security, the ATT estimated through kernel matching revealed that the main livelihood security score of beneficiary was 48.0 point, while that of non-beneficiaries was 37.02, yielding a mean difference of 11.07 with a statistics of 3.35. This statistically significant difference indicate a clear positive impact of NRLM participation on the livelihood security of rural women. Further, the unmatched sample also showed a positive difference of 8.29 T statistic, but the matched analysis provide a more reliable estimate by controlling for treatment differences. Hello?
Yeah, go ahead. We are hearing.
Yes, sir. After post-matching, a regression analysis was carried out to examine the socioeconomic factor which are influencing the participation in NRLM, where participation status, beneficiary was given 1 and non-beneficiaries were given 0, was treated as dependent variable. The result indicated that only a limited set of variables significantly influence program participation, suggesting a relatively focused targeting mechanism. Under this scheme, among the variables, the caste exhibit a positive and statistically significant influence on the participation. Similarly, type of house, material possession, agriculture, land owning, income, social seeking behavior, economic motivation, achievement motivation, and risk-taking motivation were also shown program. Now, coming to conclusion, the impact analysis using PSM method clearly established the participation in NRLM. The dimension-wise analysis of livelihood security highlighted that the food, economic, and health security showed significant improvement among beneficiaries, while educational, social, institutional, and infrastructural dimension exhibited a positive but non-significant gains. The regression analysis demonstrated that type of house, material possession, agricultural land holding, monthly income, social participation, and information-seeking behavior and other were the most influential determinants of the livelihood security. These findings emphasize that the interdependence between the tangible assets and psychological environment and social institutions support in enhancing the overall well-being of the rural women. Thank you.
Okay. Any question from online participant to Miss Pooja? Yeah, please.
डॉक्टर पूजा आपने कौन सा रिग्रेशन एनालिसिस यूज किया है हेलो
Sir, multiple regression analysis.
how you can use caste and marital status as an independent variable when these are just classificatory variable na
Sir, I have not attained the relationship between the independent and dependent variable. I have just examined which factors influence the participation in this.
do you think these type of variable are suitable for regression analysis especially for
Sir, I've also done the relationship between the dependent and independent. I have not included caste and family size in that. It is just after the matching, after PSM, I have carried out this regression to show the determinants which affect the participation in the NRLM. I have not included this in hypothesis testing.
but your result in regression analysis there was two variable caste and marital status these type of variables are not suitable for linear regression okay
Sir, I know this. I have not included that in my final result of hypothesis testing.
Okay, fine. Thank you, Pooja. Actually, it is a very good field-oriented research. Maybe you should have used some more statistical tools to express the results.
Sir, this is a part of my study. I have included other thing also in my study.
But you have not presented here?
Yes, sir. Sir, it is one objective, the main objective.
Okay, some descriptive analysis, all those things you should have included. Why this caste is having a significant impact? Means which caste is lower adapting or which caste is more adapting? Is it do you have any idea?
So it is mainly the inclusion of the caste in the program. That is, it mainly use the PIP participation of the poor method to include, in which the general category can also be come in the those poor category for participation in this scheme.
Okay. She took around 120 plus 100. Sufficient number she took. Okay, thank you, Pooja. It is really very good work. Actually.
Thank you, sir.
Thank you. So, who is left out now? Vicky, can you identify yourself? Who is Vicky?
Yes, there are two names, Vicky, Bidisha, Sarma. Your names are not enrolled. If you are going to present
Are you
today,
only a participant or a presenter?
kindly confirm.
Okay.
Okay.
So anybody who has not presented so far?
Yes, Banot.
Banot also is not here, no. Anybody would like to present? Vidisha presented, am I correct or she is?
Yes, sir, I am present. I am a presenter, but I have my presentation tomorrow at 3:25 p.m.
Fine. So you came as a participant. Am I
Yes,
correct?
yes.
Okay, very good. Thank you. Thank you. But you didn't ask any questions. You should ask.
And what about Vicky? Can you please confirm your name?
Vicky also may be a online participant. Fine. So I think, yeah. Shall we conclude? Or anybody would like to present who has not presented from the online? Yeah, but he could not join again. Effect of organic nutrient sources and enriched banana pseudostem sap on growth, yield and quality of green gram under organic farming. सर का भी मालूम ग्रीन ग्राम है ग्रीन ग्राम effect of organic nutrient sources and pseudostem sap ये कैसे क्या पता नहीं but topic is interesting. Not picking? Fine, no issue. From our side, we.
Hello, is this Bano I'm talking to? Yes, I'm from AgriVision. I'm calling from AgriVision conference. You were there, right, for the presentation.
Would you like to present? Yeah. Okay. So anything you would like to tell? Some of them, let it be some participants are there. You can tell also, no issue. What is our overall the session was really very good. We took from veterinary to nano and also again home science and different this one. It was really very interesting session. So some of the presentations were really excellent. Any comment you would like to make, sir?
So all the participants were really good, most of them rather, and I think 4 of them have presented the research achievements, research oriented presentations were there. And as the chairman has suggested, even if you are presenting a review paper, so there has to be your analysis and your contribution in the presentation. It is not that taking the data from the net and presenting it here, that we already know. But if you are taking some papers, number of papers, how many people have put what, and based on that some analysis can be done, and then the results of that analysis of the review can be presented. So this is only my observations. Thank you.
Thank you for the online presenter, also participant.
Thank you very much, participants. We are very much thankful to our chairpersons and rapporteurs for chairing the sessions. We are concluding this by now, and tomorrow we are having another parallel session. Rest of our participants can join over there. Thank you very much, everyone.
So overall more than 50% has presented, I think. It's good. T. You can end the session. Who is, any rapporteur would like to tell anything? Or? Fine, okay, no issue. So I have put my score here itself. You people can use it if you want to award anything, right? ठीक है?
सर
Yeah, please go ahead.
सर मैंने रिव्यू पेपर जो प्रेजेंट किया था तो अगर मैं मॉडिफाई करके फिर से प्रेजेंट कर दूं या
No need. This is for your future, this one. Whenever you are presenting in the next time, that time you
okay
can...
sir, I will improve sir.
Yeah, you can improve. It is, you have done it, no issue. We wanted some type of analysis. How many papers were there? Out of that, how many were dealing with this particular nanopesticide? How many were dealing with the another one? It is like that.
ओके ओके सर, मैंने पहली बार प्रेजेंट किया ना इसलिए मुझे पता नहीं था.
I am not getting, what is it?
That's okay, that's okay. You can contact the organizers, and if they are allowing you, you can tomorrow or day after tomorrow, you can present with another topic or some research paper. Thank you.
So, thank you. Thank you everyone. Initially there were some hiccups, but now it is really very much fine.
क्या हुआ था वो जो आ रहा था?
There were some echoes. Now it is fine now.
Just मेरे को ऐसे जानना है कौन उसने ऐसे No, that cannot be. Double application माने माने दीटा विंडो ओपन है कि थिला अच्छा सो जे विंडो बंद गोटे विंडो बंद करला पर सेटा लाइव सेशन होता दीटा विंडो कंकरेंटली केला गोटे मिनिमाइज हेतिला माने जानि पाउ तो दीटा प्रेजेंटेशन चालतिला हां दीटा प्रेजेंटेशन गोटे बंद होला पर सेटा ऑटो
So what about yourself? Introduction.
myself Dr. Harikrishna Swami. I am working as a scientist in botanical surveillance in Shillong.
Shillong. Yourself?
I am Dr. Santanu working here.
Extension
Botanical survey in India. Actually, we wanted help. We wrote a letter to SBA, Botanical Survey of India, Coimbatore.
Yes sir.
They told us to approach. Actually, we wanted.